Hi, we're continuing with life science and cell biology in a nutshell. Um, we're learning about ribosomes currently, and translation, is the which is making peptide polypeptide chains, aka proteins, off of mRNA molecules, which um, which are transcribed from DNA initially. So, codons, which are three bases on the mRNA molecule, each attract an anticodon tip of the tRNA molecule, which is bound to a particular amino acid. And each codon attracting that amino acid, the specific sequence of codons of a gene will bring particular sequence of amino acids. Therefore, a particular protein will be synthesized off of that gene, which is that section of um, the original DNA sequence, which was transcribed into an mRNA chain in the nucleus. And that mRNA came out of the nucleus and bound itself to the ribosome in order to make a protein. So we learned that there's free ribosomes and bound ribosomes. The difference is free ribosomes are in the cytoplasm and they synthesize proteins for use within the cell, while bound ribosomes, which are bound to with the endoplasmic reticulum, which is on the exterior of the near the cell membrane and nuclear membrane. They synthesize proteins for export or excretion from the cell or for use in lysosomes, which are a different organelle which break down materials. So, this is again showing mRNA and ribosomes and proteins being synthesized. Graphic. This is showing a ribosome in 3 dimension, which is a complex molecule consisting of the two major subunits, large and small. A note: a cell assesses specific genes by transcribing their DNA sequences into RNA sequences. A cell's characteristics reflect control of transcription. RNA carries the gene's information into the cytoplasm and enables it to be translated into a protein's amino acid sequence. Messenger RNA carries the gene sequence information. Ribosome RNA is part of the ribosomes. So it supports and brings together amino acids as proteins form. Transfer RNA matches specific amino acids to specific mRNA triplets or codons, enabling ribosomes to assemble proteins. To synthesize a protein, tRNA molecules carrying specific amino acids base pair with mRNA molecules. So ribosomes align the amino acids. A protein must fold into a certain conformation to function as well. After they're created, a change in a gene's nucleotide sequence can alter the gene's function and affect the phenotype, or the expressed phenotype, which is what is reflected in the function or the appearance of that organism due to various proteins being created off the DNA or the genes. So the difference between DNA and RNA: DNA is double-stranded, RNA is single-stranded. It's transcribed off the original DNA sequence. So this is what the molecules look like. Deoxyribose is the sugar of DNA. Ribose is the sugar of RNA. So there's no, so in DNA, you have here an oxygen is missing. So ribose has that one extra oxygen. The bases, one base is different. In RNA, it's uracil instead of thymine. Uracil has, let's see, two hydrogens that are missing, and these hydrogens must be probably removed for as as their as the DNA strand is cleaved to make the RNA, and could probably be used for its energy or something. So this is interesting. That's so this is an example of what the DNA chains and RNA look like, and the protein subsequently created. You have regions within the DNA, such as the promoter region, operator region, and the gene itself, which control the transcription of mRNA off the DNA. So here it's shown that if there's no lactose, you don't have transcription of any protein or mRNA. This no transcription of mRNA, and therefore no protein being synthesized or translated, because there is a repressor protein that is there that stops the initial transcription of mRNA. With the lactose, the repressor is removed, or bound and removed. Therefore, mRNA is created and protein is created. So that's one example. So that's showing how transcription occurs in the gene to be made into a particular protein. RNA polymerase, which helps with this transcription, with so the promoter region is the initial region which determines where the gene is to begin to be transcribed. Transcription factor binds the initial part of the sequence, so the promoter region, which starts the transcription. And RNA polymerase helps with polymerizing the RNA. So these are proteins in charge of transcription. And so that's what you have. And RNA, there's a messenger RNA, which takes the message from DNA to the ribosomes. Ribosomal RNA binds with proteins within the cytoplasm to form ribosomes where proteins are made. And transfer RNA transfers amino acids to the ribosomes in creating of peptides or proteins. So those are the three kinds of RNA present that are made off of DNA. This is showing what tRNA looks like. It has an anticodon binding mRNA binding site, so the triple base tip, which complements a particular triple base that are the codon regions of or triplets on the mRNA, while the other end has the amino acid accepting region bound to amino acids. So each codon brings a different amino acid. Thank you.